Everyone, my name is Kelly Klein, and for my senior capstone project, I will be talking about the research I participated in for several semesters in Dr. White's lab in the chemistry department at the University of Cincinnati. I will be talking about metal nanoneedle-based ion channel probes used for single molecule detection. So first, I want to talk about ion channels and why they're important and what they do. So ion channels are very important in cell functioning and signaling, and they also play an important role in controlling cell permeability, which means they allow certain molecules to pass through the lipid membrane and some they keep out. And they are commonly found throughout the human body, and without them, important processes could not occur. Recently, these ion channels have been used as nanopore sensors for the detection of biomolecules such as single-stranded DNA, microRNA, and peptides. So here we have a diagram of just the general setup that was used with a gold nanoneedle with a modified surface used to support lipid bilayer formation and to be able to use these ion channels in nanopore sensing. And this is surrounded by a PEG layer that contains protein monomers, and then we have an oil lipid mixture and an aqueous bath layer. And as you can see, there's the formation of the lipid bilayer that is starting to happen. And then there's an enlarged portion that just demonstrates a larger view of the protein channel and a single molecule binding. Tubes, and they're then electrochemically etched in 2 molar calcium chloride solution, and a sine wave AC potential of 12 volts with a 9.5 volt DC offset was applied. And this process takes around 2 to 3 hours. And then on the bottom right is just a general setup of how the gold nanoneedles are etched. Before I start to share my data, I would like to show how the current time traces look before and after protein insertion in the lipid bilayer. Before, there is a channel current of 0 since the bilayer is acting as an insulator between the 2 electrolyte sides. Once the ion channel protein reconstitutes in the lipid membrane, we see this jump in current due to free ion movement through the protein pore. So here are the current time traces for gold nanoneedles, and as you can see, there is a slight decline in the measurement. This decline represents a limitation of the gold nanoneedles, which is channel current decay due to double layer charging at the electrode surface. To combat this problem, I started using silver nanoneedles with the same setup in order to obtain more stable measurements. The silver nanoneedles were prepared using silver wire and glass capillary tubes, and they were then electrochemically etched in 70% perchloric acid methanol solution, and a linear wave DC potential of 1 volt was applied. And this process takes around 3 to 5 minutes. This is an SEM image for a silver nanoneedle, which just better illustrates the effects of electrochemical etching on the geometry of the nanoneedle tip. These are some of the current time traces I obtained for silver nanoneedles, and as you can see, the measurement is much more stable. Using a silver nanoneedle, I was able to measure several single molecule binding events. And as you can see, these downward currents or blocking currents represent the physical blocking of a molecule as it binds to a protein channel. In this case, it is beta-cyclodextrin binding to an alpha hemolysin. So to conclude, there are several big picture applications of this research. First, these metal nanoneedles can be applied as ion channel probes for the detection of biomolecules which could possibly be associated with certain disease at the single molecule level. Also, the nanoscale geometry of the probe tip provides a higher spatial resolution and can be employed to monitor molecule or ion flux from cells. I would also like to acknowledge Esther Khalil and Ryan White for all of their help and support throughout these past couple of semesters.